So then here we go for chapter 8, part 2. And you guys have seen a lot of these stages. Well, you should have seen all these stages with your mitosis lab. These are the images that are in your book, and if I remember right, this is actually a plant cell, but it's a highly specialized plant cell that doesn't have a cell wall. We also looked at one of those in the videos, if you remember. Okay, so while I go through these phases, I want you to try to memorize them right now. And I know some of you have already accomplished some of the memorization. But let's try and memorize them so you can recognize them really easy. I think I've said this already, but just in case you missed it or I forgot, your practicum for this semester, you will be using the microscope. Definitely more hands-on, like a lab practicum should be. And I will give you a slide for the onion root, and you'll have to find a cell that's in metaphase, anaphase, or telophase. You'll have to get the arrow on it. So you're going to need to know these, okay? And then plus you got to be able to recognize the phases for this test. And then it'll be visited again on the final. Also remind me, I want to give you guys the test prep today or get it posted today. I made some hard copies, so remind me of that. Okay, so what phase is this? It's inner phase. You can see the nucleus. Again, it's hard to discern because you can't see the membrane, but you can guess or estimate that there's got to be one because here comes another nucleus right over here. When they stained this, they were using special stain to pick up the spindle fibers and the DNA. Okay, and the same thing with your slides. So the DNA is stained what color? It's the blue, and the spindle fibers are, that pink or red or whatever you want to call it. So that is inner phase. Then we hit prophase. And the first one would be an early prophase. The second one would be a late prophase. And I think I mentioned something during the video did I talk about prometaphase? Actually, I don't think I did. We don't need to be concerned about that. Prophase is a long phase. So if you're in the field, you're studying these cells, you want to be able to tell the person, hey, it was in prophase, which is like B here. Or no, it was in late, which would be called prometaphase. So they just divided up prophase into the first part prophase, second part prometaphase. Did you hear that word on the video? Okay, so that's what it was. Don't worry about it. We're just going to group them together and call them prophase. So the nuclear membrane is breaking down, and it gets completely broken down, and those chromosomes are supercoiled. Well, I should say the DNA is now supercoiled into chromosomes. The spindle fibers are growing, and especially towards that C picture, they're trying to get actually to the chromosomes. Okay. The kinetochore is the part of the centromere where the spindle fibers connect. It's a little more trivial, but I just want to mention that to you. I don't think I put it on the vocab list, did I? Anybody get kinetochore? No. Okay. So a little higher level, you don't really need to worry about it, but it's actually where the spindle fibers connect to the chromosome, which I do want you to know is where the centromere is also. It's part of the centromere. So what's going to happen is these sister chromatids, the parts of the chromosome, the two sides of the chromosome, will need to be moved to opposite poles. So then what happens is those spindle fibers push and pull the chromosomes so that they get all lined up in the middle. And E here is showing like perfect example of metaphase, and they're already connected. The fibers are connected to those kinetochores, or right there in that centromere. And then those fibers are going to contract, ripping the chromosome in half. So you could argue that D is early metaphase, or it's definitely, you can see that it's moving towards the equator, or they also call it the metaphase plate, which is simply the center of the cell. Then we get anaphase. So now the fibers have contracted and they have ripped the chromosome apart, and now we are back to linear chromosomes. This is anaphase. Then we get to telophase. Now what else is happening right here during telophase? Cytokinesis. The cell is splitting in half. Okay? So get those other things closed. Just one tab if you're using your computers, please. Okay, so if we're going to just talk about telophase, we're going to concentrate on what? So PMAT, prophase, metaphase, anaphase, telophase, is the phases of what? Mitosis. Mitosis is the division of what? The nucleus. The nucleus. So when we're talking about telophase here, we're talking about what's happening with the chromosomes in the nucleus. So the chromosomes are relaxing, they're unwinding, so it's getting blurry again, and the nuclear envelope or the membrane is being formed to house and protect your DNA, right? For cytokinesis, though, what are you seeing that's happening? The cell is actually splitting in half, okay? And so for animals and things without a cell wall, there's actually fibers that are circling within and on the, I think just within the membrane, and they tighten, pinching it apart. But what do the plant cells have to do to split in half? What do they have to grow, or what do they have to make? A cell wall right through the middle of it. Awesome, you guys. Okay, so then you should be pretty familiar with these. And I don't know if you guys were struggling with counting the chromosomes. You guys are my first class. So did we do an okay job of getting these chromosomes counted? Did I kind of show you how to do it? It is hard, like right here. The one in metaphase. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 maybe? And again, you just got to estimate the best you can. Okay, so we went over all those. Okay, now, great, that's how a cell divides into two. But what makes it divide? Or what makes it stop dividing? That's what we call regulation. So it's really complicated, but we're trying to learn as much as we can because we're really trying to fight a bunch of diseases that have to do with cell division. And one of the main diseases that have to do with cell division is what? Cancer. And that's uncontrolled cell division, right? We're getting masses of cells. The cells are not, they don't even grow up correctly. They don't develop correctly. They start dividing even before they've become a complete cell. So they can't do their job. They're just taking up space, taking up resources. Sometimes they're smashing other areas. And then it's really bad if they start spreading and causing problems throughout the whole body. So that's cancer. So what happened is, scientists, and this is a little dated here, we figured out cyclins a while ago. So they realized that there's these special proteins that the body starts making when a cell needs to divide, and it triggers the cell to go through division. They name them cyclins because sometimes the proteins are there and sometimes they aren't. So they do this cycle. They go up and down. And what happened then is they realized it's all highly related. When this protein pops up, then cell division starts to happen. When it goes down, they stop dividing. So we have found the chemical that is telling when a cell when to divide. And if we could take that chemical out of the body, then maybe we could stop the cells from dividing. Or if we can busy that chemical, we can make it react with something so it doesn't work anymore, then we can control the cell cycle, which is very exciting for trying to tackle cancer. There's also a lot of commercials on cancer drugs right now about a different type of treatment. Have you guys noticed that? New type of drugs for cancer. where you don't have to have chemo. Have you guys picked up on this? These type of drugs we think are helping your immune system fighting the cancer cells and fight the cancer cells. Have you ever heard like immunotherapy, I think is what they're calling it? So there's a lot of new stuff going on. Another good thing they figured out, again, a little bit ago, was at least drugs that would help the symptoms of chemo. So you have chemo, which means what? Chemicals. So here we're going to put really, really bad chemicals inside your body, targeting the cancer cells so it kills the cancer cells. What's the other type of therapy we get besides chemotherapy? Radiation. Right? So now we radiate. We could put, they could target stuff, and they can radiate the areas also and kill a bunch of the cells. The problem with cancer is we're fighting our own cells, right? There's no foreigner, there's no virus, there's no bacteria, there's nobody attacking you that's not supposed to be there. It's a mutation has happened in your cells that is making them ignore the normal regulation stimulus. Okay, real quick, this is just really neat. So when you look at that graph closer, there's also these kinases that came up. And you don't need to memorize this, but just look at this, you guys, because this just shows you how amazing the chemistry is. So the people that found the cyclines also found this other molecule called kinases. And they seem to disappear. But then this other thing that they named active kinase all of a sudden appeared in the cells. They realized then through many investigations, problem solving, that's what scientists do, that the cycline reacts with the kinase and makes a different molecule, right? It's like a bigger molecule now. So we can't detect the cycline or the kinase, we just detect this new thing. At first they thought it was a brand new thing, but then they realized, no, these two are just bonding together. And so it looks different, and so it's detected as something different. So it's pretty complex here. This thing, with some energy and some of these inactive proteins, can activate the protein, and it allows the gene to get transcribed for making these other proteins that the cell needs. Over here, if you have inactive proteins, you could see that they bond together. So basically you got all these chemical reactions happening. And again, we just want to know about this so that we can fix what's broken. We could maybe we could do some gene therapy, get these genes to turn on. Maybe we can introduce these proteins in the body so that the body will do what it's supposed to. Okay, the other thing we figured out is there's these checkpoints. So when I say cell cycle, what do you guys picture in your head? Awesome, that circular pie graph, right? And so along the way, there's all these checkpoints on it. And again, they consist of proteins that detect mistakes and damages, and they put the cell into a cell cycle or rest. Do you remember what that's called in the cell cycle? It's a G what? G0. So then it's the cell will not divide. There's a problem with the cell, it should not divide. So what's really awesome is if the body detects, hey, this cell has a mutation. It needs to not divide, otherwise it's going to pass on the mutation, right, in all the baby cells, and we're making a bigger problem. So we have found these 4 checkpoints, and this picture is a little dated, so maybe they have found more. But this makes sense. Look at the chart, you guys. If there's something wrong during mitosis, if the spindle fibers aren't working right, and then we didn't get the DNA in each side separated, then we're going to have problems. So it'll just arrest, hopefully, if the cell detects that there's a problem. And then, so now G1, we're back into what phase? Besides the G1 phase, G1 is part of what phase? Inner phase. So this is like a baby cell, right? We're just starting off here. So then, if it's so cool, the cell does a self-analysis. Do I have any issues? These proteins run around trying to figure stuff out. And if there's something wrong, again, the cell will go into the G0 and it won't copy itself because there's something wrong with it. If everything's good, the DNA looks good, it can progress. And if it needs to divide, then the chemicals will start putting it into the synthesis. Okay, if it gets past this R restriction point, it's copying its DNA. But it's okay. We got another restriction site that checks it, and if the DNA didn't get copied correctly, it can arrest the cell. If everything looks good, it keeps going. Now it has double the DNA because it's went through synthesis. And now it's going to grow some more before it splits. It needs more organelles, it needs more size before it splits. That's the G2. Everything's good, we move and we do PMAT and mitosis, and we have 2 baby cells. They call them daughter cells if you want to be picky. Okay, a little bit more on cancer here, some vocabulary. So cancer is just mutation in the genes that encodes these proteins that lead to uncontrolled growth or uncontrolled cell division. It means the same thing. We know, you guys, this is not a guess. We know this has been proven. Okay, time after time after time, we can watch the DNA get a mutation in it from things like smoking, radiation, and then viruses. This is the newest thing we're starting to understand, that viruses can change DNA to cause mutations. So standard cancer treatments, to kill all the cancer cells, they also have to kill a bunch of healthy cells. So this is a picture of melanoma. It's a deadly type of skin cancer. It doesn't always kill the people, but it's a very scary one. Okay, and it'll spread. But most skin cancers are not melanoma. Most cancers are not malignant, which means that they, I said that wrong. Yeah, no, malignant versus, what's the one that doesn't spread? Malignant spreads. And then this one is helpful to remember because what's mal mean in the Latin languages? Bad, yeah. I'm going blank on the other one. Oh, benign. Benign means, and a lot of skin cancers are benign. They're like little out of control mold, for example. But if you see things like that changing shape, you need to get it looked at. When you go to the doctor, sometimes the doctors are busy, right? They got somebody else coming. Doesn't matter. Ask them. They'll do it. Sometimes they just need to be reminded. Or maybe they don't realize you're spending a lot of time out in the sun and you should be checked. But don't be shy. Let them look at your everywhere. And even still, they'll make marks on their chart. Maybe they're just going to watch one, right? And they'll measure it. So be preventative. Okay, so the other thing we realized here is that most important internal factors like those kinases and those cyclines, they're telling the cell what to do. Those things are made by the cell when certain external stimulus happens. So you guys cut yourself, and you'll start to see, you can see the tissues growing back, covering the wound, right, replacing the cells, repairing yourself. Once the cells get in contact with each other, and sometimes they don't line up just right? This is how we get these little scars. They're growing and then they, come together. They stop. So we know that most cells, when they start duplicating, if there's a gap, an opening, you could do this on a petri dish, once they hit each other, they stop making the proteins that make them divide. So the problem with cancer is it ignores this stimulus also. So it doesn't care that they start piling up on each other. So the external factor then is touch, when they touch each other. Okay, so I think we talked about all that. And I want to show you this picture. Sometimes it's hard for us to understand. So what? We got a growth. Why is that doing damage? It's just this little tumor thing. So what happens is, like I said, it starts taking over an area, and they don't look like the cells that are doing their job because they divide so quickly. They're not doing anything except dividing, and that takes resources, so it's going to kill the other cells around it because they're being deprived. And then what happens is it keeps growing and growing. Eventually, it's going to run into some sort of vesicle. You have blood vesicles, you have lymphatic vesicles, and once it hits this stream, it's going to be pushed somewhere, and it can get lodged in some other place and then continue to grow there. And then you'll get these tumors all over the place. That's called metastasized when it has spread to other areas. Okay, look